Assalamualaikum warahmatullahi wabarakatuh. My name is Siti Nursuryani binti Hussein with metric number 206079. Today, I will do an experiment on measuring the rate of photosynthesis. Photosynthesis is a process that provides nearly all the energy and organic carbon that is required by living organisms. Photosynthesis in the leaf of plant can be divided into two stages, which is light reaction and Calvin cycle. In light reaction, it requires a continuous supply of sunlight and occur in the thalocode membrane. The light that is absorbed by chlorophyll will be converted into a chemical energy in the form of ATP and NADPH. Hence, water molecules are also converted into oxygen through this light reaction. As for Calvin cycle, it does not require light and occur in the stroma of plant. Calvin cycle will use ATP and NADPH that was produced from light reaction to fix carbon dioxide and produce glucose. The material needed for this experiment are beaker, sodium bicarbonate, 10 ml syringe, distilled water, fresh leaves, and also a paper puncher. First of all, prepare a control sample that contains only water and also 3 different concentrations of sodium bicarbonate solution, which is at 1%, 2.5%, and 5% weight over volume. To prepare 1% weight over volume sodium bicarbonate solution, I dissolve 1 gram of sodium bicarbonate into 100 ml of distilled water. And same goes to 2.5% and 5% weight over volume sodium bicarbonate solution with 2.5 gram and 5 gram of sodium bicarbonate added to each beaker. Next, prepare 20 small pieces of fresh leaves using paper puncher. We are going to use 5 small pieces of fresh leaf for each beaker. Take the 10 ml syringe and put 5 small punch leaf inside the syringe. Fill the syringe with distilled water half through and you shall see the leaves float. Take out all the air from the leaves by sealing the opening with your finger and pulling the syringe to create a vacuum condition. Push all the air out from the syringe as much as you can. Do it several times until all the leaves sink. After all the leaves sinks, make sure there is no air bubble on your leaves and syringe wall. All syringes containing the small punch leaves and water are transferred to beakers containing sodium bicarbonate at different concentration and also to the beaker that contain water solution only. The beaker will then be placed under a dust lamp at range of approximately 30 centimeters. Observe when the leaves start to float and record the number of floating leaves at the given time intervals. As for the results, there is no leaf floating for the control samples and there is 2, 4, and 5 numbers of leaf floating for 1%, 2.5%, and 5% weight over volume of sodium bicarbonate solution respectively. From the graph, we can conclude that the higher the concentration of sodium bicarbonate used, the higher the rate of photosynthesis occurred. This is indicated by the numbers of leaf floating. The sodium bicarbonate that is added into the water will supply carbon dioxide and the bicarbonate ion will cause the leaf to sink because it acts as a carbon source for the photosynthesis. Hence, the light source, which is the table lamp, will provide the light energy for the thalocode membrane of the leaf to carry out light-dependent reaction, producing oxygen into the interior of the leaf disc in the form of air bubbles. The air bubbles around the leaf disc will create a buoyancy which then helps the leaf to float in the solution.